So in this video, we're going to be looking at factors that affect how enzymes function. So when we talk about enzymes functioning and we look at it in terms of graphing, we usually graph the rate of the reaction. And so how much enzyme or how much substrate is present can affect the overall rate of the reaction. We also have temperature, pH, and salinity can also influence enzymes and how they work. And then we have molecules called activators and inhibitors that also can influence enzymes. Okay, so when we look at enzyme concentration, so on the y-axis we see we have reaction rate, and along the x we have enzyme concentration. And as the enzyme concentration increases, we move towards the right. So right here would be like a high amount of enzyme, and over here would be a low amount of enzyme. All right, so we can kind of see here that initially, as you increase the amount of enzyme, the reaction rate also increases, but eventually it begins to level off. So let's go ahead and see why. When we look at enzyme concentration, so let's pretend these yellow dots here are substrate. As the amount of enzyme increases, the reaction rate increases. So as we increase the amount of enzymes, we increase the likelihood that a substrate will collide with an active site. So here you can see on this last enzyme that appeared, it just so happened to make connections with the substrate. So as you increase the enzyme, you increase the likelihood that it will collide with a substrate molecule. So eventually, though, it doesn't really matter how many enzymes you add. You can keep on adding and keep on adding enzymes, but if there's a limited amount of substrate, it's the substrate that becomes a limiting factor. And that's why you can see the graph will eventually level off. So it gets to a point where you could add 100 million enzymes, but if there's limited substrate, it won't matter. So here we have some substrate, and as you increase the enzymes, you increase the reaction rate, but eventually over time, it doesn't matter how many enzymes you add, the substrate is the limiting factor. Okay, so how is the rate of the reaction related to the concentration of the enzyme? Let's go ahead and then why does it eventually level off? Goes in your box 4, your box 4, sorry. Okay, so the same kind of concept when we look at substrate concentration. So as we increase our substrate, the rate of the reaction also increases. So you can see here in the picture you have red enzymes and yellow substrate. And right now there's a small amount of substrate but sufficient enzymes. Well, as you increase the amount of substrate, you'll increase the likelihood of a substrate and an enzyme coming in contact, and the enzyme furthering a chemical reaction. So you can see as the substrate increases, the likelihood of the enzyme catalyzing that reaction also increases. But eventually over time it will level off because all of the active sites are working as fast as possible. And it reaches a maximum rate. So it doesn't matter how much substrate you add if there's a limited amount of enzymes. Okay. So here, if you have just a little bit of substrate, you get a little bit of product, but as you increase the amount of substrate, you also increase the amount of product. That was probably on a timer. Okay. All right. So how is the rate of a reaction related to the concentration of the substrate? And why does it eventually level off? You can use a graph or words to summarize your answer in box number 5. Okay, so here we have temperature. Now temperature also influences enzyme function. And you can see in this graph, if we look at it, and we look at, well, what is the optimal temperature for this enzyme, we see it's 37 degrees Celsius. Now our body temperatures are 37 degrees Celsius. It could be an inference that this is an enzyme that functions inside of humans. So we learned in class about protein structure and how it folds up based on primary, which is the order of the amino acids, You have the secondary structure, which is the hydrogen bonds, creating a alpha helix or a beta pleated sheet. And then you have your tertiary structure, which is going to be greatly responsible for the shape of that protein. With covalent bonds of disulfide bridges, you have hydrogen bonds, ionic bonds, etc. This tertiary structure of a protein that is dependent on its bonds. And then if there's a quaternary structure, that would be multiple polypeptides coming together. Anyway, so here you have an enzyme that has folded up into its shape, and shape determines function. So we can see right here you have an active site. So we have the enzyme folded up, and you have the substrate come in and fit very specifically to an active site. And then, but let's think about what happens. As you heat up and increase the temperature, you increase the movement and the movement of the bonds, and so you have bonds begin to break. And as the bonds begin to break, because of the increased heat, what happens is the protein unfolds, and this is called denature. So this protein here, as the temperature increased, oh my gosh, it basically kind of unravels, the bonds break, and you have what's called denature. Now, I don't think I have that word show up here, but it's D-E-N, oh my gosh, A-T-U-R-E, denature. All right. So now this enzyme denature and loses its shape because shape determines function. And so now this enzyme no longer works, and that's what happens in these hotter temperatures. You can see here, you're not going to have any working enzymes. So temperature is going to denature the protein. Each enzyme works best at a specific temperature. So you have, like I mentioned earlier, how humans have a body temperature of 37 degrees Celsius. That's the temperature our enzymes work best at. And now when we increase the heat, though, past that optimal temperature, we increase the energy level of the molecules, and this disrupts the bonds between the enzymes and then also the substrate. And so when we look at this, it leads, there's the word denature. So it's going to denature or lose its third 3D shape, its third structure, tertiary structure, and therefore the enzyme will no longer function. In cold temperatures, it doesn't denature, but everything is moving much more slowly, so the chances of collision is less likely. Okay. So when we look at a graph of this, the optimum temperature in humans would be 37 degrees Celsius, but if you take a extremophile, like an archaean that lives in like the hot springs of Yellowstone, it's like boiling mud baths and boiling water. You have literally boiling streams. These organisms are going to have temperatures at a much higher level that they work best at. And as luck would have it, we actually will use some of the enzymes from these organisms in DNA like technology and biotechnology. So kind of cool. We'll talk more about those later in the school year. All right. So when we another thing that can influence enzyme function is pH. So here we see a graph, and we can look at pepsin and trypsin, and then also an enzyme that's red, right? So we can see that each of them have a very specific pH that they work best at. Now, if we think about what does pH stand for, pH is the power of hydrogen. So the more hydrogens you have, the more acidic the environment is. And when we think about a hydrogen ion, we're looking at an H+. Now just think, what would an H+, what kind of bonds would that interfere with? So if you increase the amount or decrease the amount of hydrogen ions, you potentially are interfering with hydrogen bonds and ionic bonds, which would affect the secondary and tertiary structures. So when we look at pH, the pH is either adding or removing hydrogen ions, and this is going to disrupt the bonds and the attractions that form that tertiary structure. So when you have hydrogen bonds and ionic bonds forming that tertiary structure, now if you change the ionic environment, now you have a disruption, and now it may lose its shape. denaturing the protein. So different areas in our body have different optimal pHs. So each place in the body having a different pH, we can see that, for example, with enzymes. So when you eat your food, your enzyme called salivary amylase is responsible for breaking down your starch. And so if you have some French fries, you have these potatoes made of starch, your mouth waters, there's enzymes in there. It's going to break down the starch, the bonds between the glucose molecules. But then when you swallow your food, your stomach is incredibly acidic, a pH of 2. So that enzyme, salivary amylase, will denature in your stomach and no longer function. But then you have enzymes called pepsin that work in your stomach to break down proteins. There's peptide bonds between two amino acids. So pepsin will break that peptide bond by hydrolysis and break it. But now when that food is in your stomach and it goes into your small intestine has a very basic environment, a higher pH. So there's pH buffers that are released from your pancreas that go into your small intestine, and then those enzymes will help to break down and digest fat. So pepsin that works best in an acidic environment in your stomach will be denatured in your intestines. So each place has a specific pH, so their enzymes work best in those locations of the body. And again, the pH will affect enzymes by influencing its overall shape because the pH with the hydrogen ions will interfere with the secondary and tertiary structures of that protein. All right. So another factor affecting enzymes would be salinity. And in salinity, we're looking at salt concentration. And this is the same principle that has to deal with pH, with the changing in charge. Ions, cations or anions interfering with those ionic bonds, hydrogen bonds, disrupting that overall interactions between the amino acids, forming that secondary and tertiary structure. It can lead to disruption of that protein shape and denaturing it. Where this would matter, or one example, you could think about like ocean salinity. Think about like the melting glaciers, for example, adding fresh water into the environment, changing the salt concentration for little invertebrates that live in the ocean water. Or you can talk about humans either adding too much salt or becoming dehydrated, etc., within our own bodies. All right. Okay. So let's go ahead and think about what do they have in common. Temperature, a change in temperature, a change in pH, a change in salinity. What do they have all in common? Like, what do they do in terms of denaturing proteins? All right, and that gets to be summarized in your box number 6.